All right, let's get down to business and work on understanding how the genetic information, the genes in our chromosomes, are used to direct the production of proteins out in the cytoplasm on a ribosome. So, first part is transcription, getting that genetic information in the DNA out of the nucleus in a form that can go to the cytoplasm and carry that information to help build a protein. This process is called transcription, and that term means writing across, literally. Transcribing DNA into RNA, I like to think about it like changing fonts in a word processing program. The words might not look the same in a different font, but you can read them and you know the meaning just the same. And RNA is not exactly the same as DNA, but the information in the sequence of DNA, and if we look up here at this sequence of DNA with the bases ATC, ATC, CAA, etc., that sequence is preserved. That information of that sequence is preserved when we transcribe an RNA using the DNA as a template, because the RNA nucleotides are complementary to the DNA nucleotides in the same way that the DNA nucleotides are complementary to each other. So in the same way that we can use one strand of DNA to know what the sequence is on the other, we can look at the sequence of RNA and know what the sequence of DNA bases was. So that information is preserved and can be carried out of the nucleus to the ribosomes in the form of one of these messenger RNA molecules. So recall from the previous video that we noted a small but important difference between RNA and DNA. RNA nucleotides do not have the base thymine, the T. Instead, they have a very similar base called uracil, and the part of the base that interacts with other bases to form a complementary base pair, to hydrogen bond, in other words, the part of the base that does that is circled on these diagrams. And you can see that U and T in this really important part of the molecule are identical in structure. So in the same way that thymine would be complementary to and hydrogen bond to an adenine, a uracil base would do exactly the same thing if it's RNA instead of DNA. So this process of transcription, thankfully, unlike DNA replication, which has lots and lots of different enzymes doing lots of different functions, transcription is done completely by one giant enzyme complex called RNA polymerase. RNA polymerase does the whole thing. It unzips the DNA, it lines up the nucleotides, or makes sure that the nucleotides line up appropriately, and binds the nucleotides together to form a single-stranded messenger RNA molecule. The RNA polymerase enzyme knows where to bind on the DNA because the regions that are next to genes have sequences called promoters, and those promoters are sequences that RNA polymerase recognizes as being, hey, jump on here, this is where you want to transcribe a gene to get that information. So similarly to the process of DNA replication, you have to unzip that DNA so that the nucleotides are exposed, the bases of the nucleotides are exposed, and can be used as a template to build that RNA. A rather inconvenient fact about RNA transcription is that we have discovered that the genes in our DNA have a lot of junk in them. And parts of each gene that is transcribed into a messenger RNA to eventually help build a protein, parts of those genes are segments of DNA that don't code for the protein. And those parts are called introns, which is short for intervening sequences. And the introns have to be removed so that we get the correct recipe for building a protein. And that process happens inside the nucleus. There are little structures that can bind to RNA and chop out the bits and then splice together the parts of the messenger RNA that actually do code for protein, and those are called the exons. And so to make that sequence on the messenger RNA the correct code for building a protein, we have to chop out these introns that are in the middle of the pre-messenger RNA. All right, so how do the A's, T's, G's, and C's of DNA converted to the A's, U's, G's, and C's of messenger RNA? How are those used to build a protein? This is the second step. This is translation, where we are swapping from the sort of language of nucleic acids to a different language, the language of amino acids in a sequence of a protein. So DNA and RNA are sequences of nucleotides. Proteins are sequences of amino acids. And the genetic code is how we get from the nucleic acid sequence to the protein sequence, the amino acid sequence. We gotta translate from that one language to the other. There are 20 amino acids that can take part in the production of a protein, 20 different amino acids. And there are only 4 nucleotide bases. So we can't have a one-for-one, one base for one amino acid code. Instead, it takes 3 nucleotide bases to form a sort of word that signifies a particular amino acid. And those 3 base sequences on a messenger RNA are called codons. So as we look down the length of a messenger RNA, every 3 bases in sequence represents the code for one amino acid. AUG means the amino acid methionine. AAC means a different amino acid. And here's how you know which amino acid is signified by each triplet, by each codon. This diagram, there are lots of other diagrams and tables that you can use, but I think this one is the simplest to use. You take the 3-letter codon in the sequence of messenger RNA, and you follow from the inside out on this diagram to determine what the amino acid would be in that particular spot in the order from the gene. So if we have the codon on a messenger RNA, the 3 base sequence AAC, we start in the middle with the A, the second letter also A, so we go out to this section, third letter C, so we go out here and we see that the correct amino acid for that codon is asparagine. And good idea to practice. I've given you 8 codons there. Use this wheel diagram to help you determine what amino acid would be coded for by each of these codons. And so keep in mind that that sequence on this messenger RNA that determined GAA was originally a sequence in DNA back in the nucleus. And translation, this final step, where the codons on messenger RNA are translated into a sequence of amino acids. This is where we finally get the ribosomes doing their work. And it could be a ribosome out in the cytoplasm, a ribosome on the rough endoplasmic reticulum. Ribosomes are ribosomes. And here's how it works. We have our messenger RNA that has a sequence of nucleotides complementary to the gene back on that chromosome in the nucleus. So the information from the gene is carried out to the ribosome in the form of this messenger RNA. The ribosomes grab messenger RNA molecules, and specifically they grab at the sequence AUG, which is a start codon. So the ribosomes know where to begin translating the messenger RNA into a protein. And so the ribosome jumps on there, grabs at the start codon, and starts reading the 3-base sequence one after another. And it does that with the help of these transfer RNAs that we mentioned in the last video. Recall that transfer RNAs are also molecules made of RNA. They have RNA nucleotides that can be complementary to other RNA nucleotides, and each transfer RNA molecule has a unique amino acid that it can bind and carry. In the case of this transfer RNA that has the sequence UAC in this region called the anticodon of the tRNA, that particular transfer RNA carries methionine and only methionine. So everywhere there is an AUG on a sequence of messenger RNA, this transfer RNA would show up with a methionine in tow. Likewise, every place there is a UUC on the messenger RNA, the transfer RNA with the complementary anticodon AAG would show up and bind to this spot. And that transfer RNA, the one with AAG as the anticodon, always carries the amino acid phenylalanine. And so everywhere there's UUC on the messenger RNA, phenylalanine will be the amino acid that goes in that spot. Next codon, AAA, the transfer RNA that would be complementary to that would have the anticodon UUU, and that particular transfer RNA always carries the amino acid lysine. So as we go down the messenger RNA, transfer RNA, tRNA after tRNA jumps on, binding to the complementary sequences on the messenger RNA, and carrying a specific amino acid to add to the chain in a growing protein. So further down the line, we continue on. The amino acids are bonded to one another by enzyme structures within the ribosome. And the ribosome progresses right down the messenger RNA until it has attached all of the amino acids to one another. Eventually, the ribosome will get to what is the end of the messenger RNA, the end of the coding region of the messenger RNA, and that's denoted by a stop codon, a 3-base sequence on the messenger RNA that tells the ribosome, whoa, this is the end of the line. And when the ribosome gets to that stop codon, two things happen. The ribosome detaches from the messenger RNA, and it releases the chain of amino acids, which is called a polypeptide. Polypeptide is another name for a protein. I wish we could just stick with one term in biology. And so that's how it's done. The DNA is made up of a sequence of DNA nucleotides in the nucleus, and the information is that sequence. The information carried in our genes, in our DNA, is the sequence information of the nucleotides. That sequence can be transcribed into this different font, so to speak. Messenger RNA, a very similar molecule that would be able to carry that sequence information out of the nucleus to the ribosomes where proteins are made. And then that messenger RNA data, that messenger RNA information, is translated into the sequence of amino acids in a protein, with each triplet, each codon, being specific for a particular amino acid in a protein. That's all, and that's a lot. Here are some questions for you to think about. If you understand these concepts well, these are questions you should be able to answer. And if you don't understand anything that we've talked about, or if you're just a little fuzzy, or if you just think you understand it but you're not sure, bring your questions. Let's talk about that.